everybody. Welcome to session 20. It's a Q&A session with Montana State University's Barley, Malt, and Brewing Quality Lab. It's going to be a brief update on current work being done by the lab, including research, analytical services, and also how the lab just generally supports the craft malt community. The lab's headed up by Hannah Turner, and she's also a extremely high-performing guild board member, I'll add. And her lab services, the broader MSc breeding program, which is headed up by Jamie Sherman here, and then Joe's a PhD candidate studying plant genetics. And together they work to improve barley of all types with a focus on malt barley, and as well as supporting the region by providing barley and malt testing and also beverage testing for farmers, craft maltsters, and brewers. I want to thank MSU for their silver sponsorship of the Craft Malt Con 2021. And as part of this contribution, they'll also be giving out a voucher for some lab services as a door prize. So more info on that to come. And I also want to highlight the importance of the Guild's early investment in getting out in front of malt quality concerns with all of this testing capacity, which the Guild's early investment really helped launch labs like Montana State Universities and Hartwick's and Colorado State Universities now. So just kudos to the Guild founding members for that. And without further ado, I'll pass it over to Hannah.
Thanks, Bill. So to kind of orient you all to the talk that we're going to do today, we're going to cover a whole bunch of topics. We're going to try and do it in 20 minutes. So mostly it's just going to be mentions to various projects that we've got going on. Our hope is to generate some questions out of this and kind of let you guys direct where our conversation will go. And with that, so the voucher that we're going to be giving out today, after the talk, Jamie, Joe, and I will go back through the questions and we'll pick the one that we think is our favorite. We'll reach back out to that person and offer that voucher out. So with our first slide here, to kind of frame our program, we go by three pillars. So research, service, and education. With that, we've got a lot more information education-wise and resources available through our website. So I encourage you to all check that out. In our vendor booth today, we also have a virtual lab tour. So welcome to go and join us for that. And also just follow us on social media. We've got lots of knowledge that we're trying to bring for barley and malt, all that knowledge to everyone. And with that, I'll hand it over to Jamie Sherman.
Thanks. So I just wanted to give a brief introduction to what we're thinking about and working on in the breeding side, and then talk a little bit about genetics research and the work we're trying to do on quality. So my focus very much is on the research that we're working on. Hannah, could you go to the next slide? So that picture was some of our Montana fields that where we actually do our testing. The previous slide was the picture of the group. It's kind of jumping around. But anyway, that's okay. And it's okay. And then there we go. Okay, so here's our group. And everybody's not there, but you can see, look at all the undergraduates we have working for us. All this stuff we do is very labor intensive, both in the field and in the lab too. So it takes a lot of people, and all those hands make all the work possible. Okay, ready? And then I did have a slide thanking our sponsors. We do really appreciate our sponsors, and don't go back though, Hannah, it's okay. And I'll talk about a couple. Our probably biggest sponsor are actually the growers of Montana. So a big focus for us is developing new varieties. And of course we need the varieties to work for the growers. And so that's really one of our first concerns. But then we also want our varieties to work for the end users, the maltsters, the brewers, if we're talking about malt barley. So we did do a recent release of a variety that we've named Buzz. It's actually named after a grower from Montana that's been extraordinarily supportive of barley research. And it's actually a daughter of Hockett. And the improvements are it has a low protein gene combined with a gene from Hocket that makes it have stable plumps across environments, especially dryland environments. And it also has a faster hydration than Hocket, so it could be an advantage to maltsters going through that early germination steps faster. It's a good thing for growers because it has stable malts proteins, so, and it's shorter in stature, so it has less lodging. So that's all good for growers to be able to make malt quality. And so we're excited about this variety, and hopefully eventually, if you wanted to, you could have access to it and malt it. Okay, next. Another thing that we're working on, I just want to mention, we are interested in developing winter barley. Winter barley has not been possible in Montana because it's too cold, and today I really believe it. We're getting good, hard selection for cold tolerance today. It's minus 16, and so it's normally too cold in Montana for barley to survive the winters, but we're trying to develop cold-resistant lines by using material from a special collection from a guy named Vavilov who lived prior to World War II, and he was a Russian. He traveled the world developing and getting winter varieties, and so we have access to that, and we're hoping that that will give us enough cold tolerance. So our long-term goal is to have malt barley that can survive the winters, but we're a long way off. Okay, next. Oh, there was Vavilov. So another thing we're working on is we also want to select for good root systems. So this is to try to make our barley more adapted to dryland conditions, which means that, of course, my phone's ringing. Sorry about that. Just ignore it. So it is the root system we normally don't select for. But if we have a better root system, then we're going to do better with drought conditions. So that's something we work on. Next slide, Hannah. The problem is that the root system is hard to select for, like you can't get down underneath the ground. And so we've developed a new system where we can take photographs of the actual plants with a drone. The camera is the red picture. The drone you can see is the helicopter looking thing, and then the pictures it generates are next to it. The green pictures is taken with a color camera. The picture above it is taken with a camera that's actually measuring different levels of light reflectance from the plants, and that can give us information about how good the root systems are, which is kind of crazy, but it's true. And so what we're doing is we're trying to improve our selection for good root systems through this process. And New Belgium has been critical in providing, they helped us buy this better drone, and they've been supportive of this research. Next slide. And so now I'm going to turn it over to Joe to talk about his research specifically.
All right, thank you, Jamie. So I've got kind of two major research focuses. The first, in kind of understanding genetic controls of malt quality, is understanding speed of hydration at the endosperm. And the way we measure this is through the Chapon test. We also look at dormancy and then pre-harvest sprouting. And we've got data up in front of you here that is from some elite crosses for a biparental population, where what we're trying to do is find these hydration genes and stack them so that we have fast hydrating grain with some dormancy that's resistant to pre-harvest sprouting. Just a quick read down. That we've got speed of hydration. Our dormancy is measured how many days after harvest it took for the grain to germinate. And then our pre-harvest sprouting measurements, we were measuring alpha amylase in the grain, and anything over 35 is considered severely damaged. Go to the next slide, Hannah. The other thing I'm working on, and this slide ended up being a little smaller than I thought it'd be, but is I work with an heirloom population where we're looking at all sorts of different malt quality traits to see maybe what we've lost, as well as looking at metabolites and trying to look at some flavor profiles. And what we have here is what's called a Pca chart, where we've taken all of the malt quality traits that we've measured for each individual line and compressed them down. So each one of these points is a line, one of our experimental lines. And the way you read this is the center of all these vectors is the mean for any one of these measurements. And if you follow up and to the left, it's extract. So lines that move up and to the left along that extract vector have higher extract. And if they move down and to the right, they have lower extract. And in this case, it lines up with beta-glucan as well. So yeah, and then with tables like this, we can then work to overlay metabolites and see relationships between these metabolites and malt quality traits that we care about. And yeah, with that, I'll pass it on to you, Hannah.
Talk a bit about some of the work that we do out of the lab. So with that service and education aspect, we do quite a bit with doing lab tours and educational events, doing wort tastings. We've got an image there that is us in the field at one of our field days doing wort tastings to highlight varieties. We support the breeding program via in-house malting and quality testing. We're now brewing out of the lab as well. And we work with clients that are all over the Northeast and even all over the world. So that map up there, everything that's in blue, we've got clients from a pretty wide region that we're working with now. And then all of that knowledge we're trying to share as well through various social media platforms, trying to make lots of people more aware of what barley and malt is and why that's why we say no barley, no beer. A few of the projects that we've got currently and on the horizon, we've got some projects with various different groups that we've paired up with. The first up is our Brewers Association trial, where we're looking at variety and level of modification and how that interacts with the kilning to create flavor. That's a project that we've paired up with CSU on, and they're helping us with the metabolomics and doing some sensory panels and trying to tease out how those interactions impact the malt itself, but then also how that may translate into the beer. Additional flavor aspects that we're looking at, we've got a student from Brazil, Aline Brito, who hopes to come this summer, which hopefully, you know, with COVID and all of that, hopefully that is able to work out, hopefully at some point, if not this summer. But she's planning to come and do some work with koji malting and altering grain through koji fermentation. We're also looking at a project, so this is a project with Aaron Justice at Ballast Point, where we're looking at the impacts of terroir and how that can impact the ionic composition of wort. That could be important for brewers because it would impact their process, but it would also potentially have impacts on final flavor. So we're pretty excited about how that project is coming along. We've got, in partnership with the Montana Wheat and Barley Committee, which is one of our major funders, a project to bring this variety selection tool, and that would make it so that there would be an interactive tool that you could go in, maybe select either a certain year or a certain variety or a certain location across Montana, and it would aggregate all of the data that we have on that different variety, and it would just be a tool for growers to be able to go and identify what line or variety might work best for their particular situation. We've got a project in collaboration with Hartwick and Aaron McLeod there, looking at GN management. And so this is kind of a dual-sided project. One aspect of it is looking at varieties that are commonly grown in North America, which we've only got a few available currently that are non-GN. And as that's such an important thing for distillers, we wanted to Although breeding programs are working to create new lines that have that non-GN trait, there's not a lot of options now. So looking at ways to manage GN levels in the current varieties, kind of in the interim, that paper is also going to go into best management practices, both from the grain side and the malting, but also into the brewing side, or into the distilling side. And then lastly, we've got projects coming up where we're interested to look at sprouted grain, food barleys, and value-added products for the malt house. Something to bring some stability and diversification to all you maltsters. And then lastly, we do offer the majority of the testing that we do through our program to support the breeding program. We do also offer that as a third-party service. This is just a smattering of the things that we're able to do through the lab. All of our packages, pricing, all of that are available on our website. So we encourage you to check it out. And if you have any questions, make sure to reach out to us. And so with that, I will open it up. Our contact emails are there, but we'd love to answer any questions and see what you guys are most interested to hear about.
Well, thanks, Montana State University Quality Lab. That was very interesting. You all are providing a lot of valuable services to the community. It looks like we have a few questions here in the question box, so we'll get started on the top. We have what work is going into bringing back or improving heritage barley varieties? How is that process initiated?
So what we did is we kind of screened our germplasm repository, which is actually in Idaho, and we pulled out lines from that. These lines have already been genetically characterized, so that's a big advantage for us. So we have that, what we call genotyping data. That just means we know something about the genetics behind the lines. So then what we've been doing with those lines is we've been growing them and testing them, malting, and then we've also been sending that material to CSU, and they've been doing metabolomics on it. We've created an enormous amount of data that we're still kind of wading through. We saw so many differences that it's really kind of the can't see the forest for the trees issue. So we really are working towards narrowing that down, coming up with, we have multiple storylines that we think are going to be important. And then an offshoot of that was really this Brewers Association project where we just took a few of those lines, like Hannah mentioned, and we malted them in several different ways, and we kilned them in several different ways, and now that metabolomics is being run on that, we're doing some flavor analysis on the wort, and then we're going to pick the most divergent material and come back and brew it. We just instituted a brewing system here at MSU we're pretty excited about. So now we can combine some of our malt quality research with brewing research. So that's where we stand. We've got a lot of interesting data. Hopefully the stories will be coming out this next year.
Great, thanks for that. Yeah, this is, I love the collaborative nature of the work that you're doing on heirloom varieties, and yeah, I think what Matt Drew is getting at is he'd love to be part of the process. I think we all would in some
well,
way.
that sounds exciting. Like if we could, the more the merrier.
Yeah.
So think of a way that you would like to participate with us, and contact us. We're very, very interested in those interactions.
Fantastic, thank you. Okay, can you elaborate on the methodology for speed of hydration index? Devil is in the details.
Yeah, I tried to get a little bit of that in the chat, but the general process is we use the Chapon test. So right at the steep out point, we will take a T-ball subsample of grain. We will boil that to gelatinize any of the endosperm that has been hydrated, and it transitions from a chalky texture to transparent if for the portions of the endosperm that have been hydrated. And then we will take from that T-ball subsample 25 seeds, take cross sections of them, and score them based on how hydrated those seeds are. They get placed into categories starting at 0, which means no hydration. Then it's a quarter, 3/4, or a quarter, 1/2, 3/4, or 1, which just means 100% hydrated. So yeah, it's just sitting there at the 25, 50, 75, and 100%.
We do have a spot on our website in our learning center that shows pictures and kind of talks through what that process is too, if anyone's interested to learn more about the Chablon test.
Great. Thanks, Joe and Hannah.
Yeah, I think that's been one of the exciting things that we think we understand the genetics now, and that paper's being put together too. So the genetics of the speed of hydration, which I think is going to be really useful to breeders and maltsters both.
Very. This is of particular interest to me. If having barleys that express stay green under drought stress, do these genotypes still express high proline? Is higher proline expression part of the stay green mechanism?
Not that we know of. The stay green mechanism that we understand right now is about delaying senescence, which is happening. I don't know how much in the weeds I should get, but genes are turned on and off by other genetic factors. And so there are genes that control how quickly something senescence goes from that green stage to the brown stage. And those genes are what's controlling stay green that we understand right now. What we're investigating is there seems to be an association with bigger root systems. And we don't really know the cart before the horse sort of thing. Is that delay in senescence also enabling the roots to get bigger? But we aren't sure about that. We think that the roots may just be bigger. Anyway, there may be a genetic mechanism also involved that affects the root development. And that's what we're really interested in. And this is kind of a big sustainability thing that has implications that are bigger than just barley. Like it would be cool for, any crop to be able to control the roots. So that's why we're really super interested in it. The proline side of things is proline is one of the important amino acids that forms the storage protein in the endosperm. And we do have evidence that we've talked about before that there are different levels of proline. We're working on the genetics of that. That's part of Joe's project too. And we've got a cool project going on right now where we grew the lines that varied the most in proline and dryland and irrigated. And the reason we wanted to do that is there is some evidence that proline might be associated with drought tolerance. And so we just want to sort of figure out if that's what's going on. I kind of don't think it is, because when we saw the differences, that all that material was in one field, had the same conditions. But we're just running through those tests and we're genetically dissecting it. And we care about proline because we think it might affect growing quality.
Fantastic, thank you. And we are a little bit over right now, so
sorry.
No, it's all good. I think this is one of those sessions where we have a little bit more time. So the last question I'll ask is kind of a little bit of an extension of something you've already said, but you have a great list. Jason Parker says you have a great list of projects. What's the best way for us to follow your trials and learn of your results? So I'm thinking what's the best way for a lot of people to follow you without having to email you directly?
Which sometimes I'm not that great about responding, but I really try hard. One thing that I would honor Hannah for is that she has done such a great job of keeping our website up to date. So that would be your first go-to. And as soon as we get a paper published, we're posting it there as well. So that is probably the most minute by minute. She's trying to give kind of weekly updates of what we're working on. She's got 2 students, a marketing student and then another student working for her to help her with that. So we're trying to, be facing outward as far as that's concerned, to get that information out as quick as possible. If you see anything on the website that you think is incomplete, unclear, want more information, want to participate with us, please do send us an email, and we will get back. Yeah.
That's a great invitation, and I'll echo the fact that the MSU website is specifically your lab's website is amazing. Hannah has done an outstanding job pulling a bunch of resources together for our craft malt community, from farmers to, from researchers to farmers to all the way to brewers. So anyways, thanks again for joining us for this vendor session here. And thanks again to MSU for the sponsorship and helping make this conference possible. Thanks everyone for joining.
Thanks so much, guys.
Thanks, everybody.